transformative and powerful from a technology standpoint. And two is, what are the diseases we want to impact? And the thinking has evolved. In the early days, people said, oh, CRISPR is all about rare diseases. Here are the mutations we know about. Here's hundreds of rare diseases we can impact. But if I project forward, I think CRISPR is going to have equal impact on rare diseases and common diseases. So we made that bet to say we're going to have a mix of both, because we're not going to say these are the diseases we want to play in. It's going to be where can we have the most impact based on the technology we have. The second is the fundamental bets to say CRISPR, obviously sickle cell and thalassemia is one franchise, and it was very clear that that was the best place for us to start off with, and we have now approved drug with Casgevy. But outside of that, we parlayed competencies. We knew cell handling, we knew cell technology, so we said we're going to go into allo CAR-Ts, or CAR-Ts in general, because we can use CRISPR to make all these modifications to make the best-in-class CAR-Ts to impact cancer. And it's going to bring a new frontier in the fight against cancer that's been ongoing for the last 60 years now, ever since we declared the modern war on cancer. Then we said regenerative medicine. This is an area that still, I think, is underinvested from a venture standpoint and everything else, because if you're able to replace organ systems, that transforms how we think about not just certain diseases, but even longevity. I went to this longevity conference where people were saying if your brain is intact and you can replace all the organs, that just you think about longevity very differently than you think about it today with telomeres and everything else. And then we have this other vertical, which is in vivo gene editing, which was, we weren't the first there, it was enabled by another company, but now I think we could be sort of the biggest mover in the space, because we're going to have many, many programs in the in vivo setting, not just for liver editing, but for other organs as well. So that's the logic of the different franchises. And then the orthogonal axis of the disease areas. And that's how we think about our entire portfolio from a risk-reward standpoint. All that said, I think, the bar for us to enter to call something a program is very high. It has to be something that really changes the disease landscape.
As you spoke about, you talked about CRISPR being a very important tool here that you can use in the future to treat all these diseases, but there's been so much innovation in gene editing. And so how are you adapting to this dynamic landscape to think about a sustainable pipeline?
Absolutely. I think one of the most important considerations for us is that we don't get disrupted. We are the leaders in the gene editing space, and we have been. But we want to make sure that we're looking at every improvement that's coming out there. And this is similar to what happened with antibodies in the 80s and 90s, late 80s, early 90s. And in fact, we're seeing antibody improvement even today. People are creating all sorts of bispecifics to give it sort of a new wind in the sails of biologics. But, ultimately value is going to accrue to the ones that use the improvements to make good drugs. You can't just say I'm doing improvements for improvement's sake. So it's innovation with a purpose. But let me tell you what we're doing at CRISPR. We have established a group called CRISPR Tx that is dedicated to all sorts of next-gen editing. And the notion of CRISPR has changed. It was an interesting experiment we did. There was an artist who had done a rendering of CRISPR for us when we started the company. And this was like a graffiti art. And the notion of CRISPR was molecular scissors. So it was like the art incorporated scissors. And today the notion of CRISPR is a cargo van that is taking effector proteins to a particular site on the genome. So with the the premise is still the same, with the 20 base pair or 19 base pair, what do you want to call it, sequence that's very specific in the genome. You can bring any effector protein, whether it's a base editor, whether it's a reverse transcriptase, whether it's a ligase, a recombinase, or whatever else, right? So you take the entire toolbox of effector proteins and you can do it, bring them site-specifically to the genome. So that opens up a lot of possibilities. But you have to be very practical and thoughtful about what you're going to apply, because, if you just start chasing technologies, you're never going to actually have a program that's you're going to move forward. So for each disease, what we're looking at to say is, for this disease, what is the best way to address that disease? And we're going to incorporate all these technologies that are available to us, and we're doing a lot of it ourselves. We're developing newer technologies ourselves, in addition to incorporating what others have developed.
Could you just touch on delivery technology in the context here and your strategy around optimizing for the different tissue targets?
Yeah. So the state of the art where we are right now, if you go back 7 years and say, what were we thinking then versus where are we now? I think the world was a lot more bullish on viruses, delivery vehicles, versus LNPs or polymers or any other modalities. And, what's changed now is what we realized is LNPs may be a better way to go because of two reasons. One is, generally, they're degradable and they're safe. Once you're done with the delivery, they're gone. So you don't have viruses hanging around. Second is, it's much more titratable than you are with viruses. So there's variability than what you may get with AAVs or lentivirus. And lentivirus have their own set of issues. And the third point is the regulators feel a lot more comfortable with delivery vehicles like LNPs because it's easier to handicap versus viruses. And so, with that sort of backdrop, we're investing quite a bit in LNP, not just for the liver, but for other organ systems as well. And we just showed data at ASGCT for delivery using LNPs to the eye, which wasn't where we were, the field was 5-6 years ago. Everyone was thinking AAV is the way to go for the eye, right? But now you can get delivery with LNPs. So it's been a dramatic shift. That said, there's still challenges. It's not solved outside the liver by any means at this point. People will say, oh, here's editing in bone marrow with LNPs and look at the great data. Yes, but at very high doses without having shown durability yet. So you still have ways to work these out where you're targeting only a certain organ and detargeting the liver, for instance, detargeting the spleen. People have shown now LNP data for the lung. People are showing LNP data for the eye, as I said, and kidneys. I think there's more work to do. But when that gets solved for other organ systems, you're going to see another wave of applications with gene editing because you're opening up a whole new opportunity set.
So let's start here with your commercial portfolio and talk about Casgevy, which is partnered with Vertex here, about the launch to date, and maybe walk us through how physician and patient feedback and uptake overall has been.
First of all, I'll say that we're very pleased to be partnered with Vertex on this important launch. Launch is important not just for us as companies, but in fact, when we were at the ARM meeting, that was the most talked about. More so than some of the PTR or DMD that people are looking at as investors, because Casgevy, for many reasons, I think the unmet need is so high for these patients. They have no other options. Here's a curative option. Can we as a system work out how a medicine like Casgevy can also be economically very fruitful? And for that, I think the launch is a nuanced launch. It's different from what people typically think of as the ways to handicap launch or look at launches. And I think what we're very pleased with is there is significant demand. And again, Vertex is going to do all the sort of guidance to the street on how to think about the launch and what the parameters are and how they're going to disclose, cell collections, for instance, or revenues. But what I'll say is from my own experience, we're deeply involved with all the investigators in the community. The demand is there. Patients are saying, this can change my life. I want to avail this technology. What surprised me is that, the system has been very supportive from a reimbursement standpoint, more so than what I'd anticipated 8 years ago, for instance. The demand is global. It's not just the U.S. And when you have a therapy like available, hospitals figure it out. There are a lot of questions and, I would say misgivings about whether the hospital's going to have enough beds or they're going to have enough staff to deal with the autologous therapy like this. And they figured it out in the Yescarta case, right? And now they're going to figure it out in the Casgevy case, because when there's an available treatment, the system, finds solutions around it. And so I would say the fundamentals are pointing in the right direction, but we have to show you how that's all working out. And, Vertex, as I said, will disclose these information on cell collections, et cetera, every quarter. But I feel very good about the Casgevy launch, and I think it's going to be, an unusual launch in the sense that it's going to be a growth driver for a very long time to come. Typically you've seen with pharma launches, you get the early pickup and then they flatten out. And here, it's going to keep growing for a very, very long time to come.
Have there been any sticking reimbursement and
without going into any specifics, just in the world of rare disease reimbursement these days, I think what you're seeing is on the U.S. side, there was a bit of this reaction to certain things that happened a decade ago, or with the rare disease pricing, and there was a reaction, but it's all normalized again. I think if people are saying if there's real solutions that are needed, this is not what's changing. Like GLP-1 is dramatically changing how they underwrite, their ability to cover medicines for employers, right? But rare disease is not changing that equation for them. And so there's, a lot more understanding of how to think about cell and gene therapies and in the U.S. and support. I think in Europe, they're still figuring it out. I think the systems are all very, customized to each country. They all have their own processes. Things, don't move as fast as some other countries. But again, there's an appreciation for medicines like Casgevy. I mean, as evidenced by the early access in France, for instance. I think there's just maybe a little timeline, but it's gonna get sorted out. And I think the rest of the world, there's enough burden and, there's significant economic growth in other parts of the world that are non-US, non-Europe, where the markets have become more attractive now, and there's enough patients that you have to take those markets very seriously. So I think, that's the great part about having Vertex as a partner that has global presence, the ability to move in different geographies, but also incredible experience with dealing with European payers. So I think, not foreshadowing anything, but Vertex will guide, but I feel good from where I'm looking at things.
And, any thoughts on how patients may be opting or physicians opting for gene editing versus a gene therapy in this marketplace?
Yeah, I think it's hard to handicap that right now because what's happened is, not every site has both offerings, right? So to get a truly controlled experiment, you have to have a site that's offering both without any investigator, or the physician bias to say, what are patients picking at the end of the day? But what I heard from one of the nonprofits that supports these patients that, we help is overwhelmingly, if they're given both options, they're going to choose a CRISPR-based option. It's hard to explain, but it's just easier for patients to understand. They say, I understand this notion of editing my gene, and I've read about CRISPR. I've seen, an NPR article, X, Y, or Z, and this seems like the way to go for me. I don't think they understand lentiviral risks of AML or MDS, and those are all things that the patients don't fully understand, or the patients don't know how to think about it. But simplistically, I think what you're going to see is a majority of these patients just opt for CRISPR if both options are available.
With regard to optimization on the forward, I think currently it could take up to 5 to 6 months to get treated with Casgevy. In addition, you've got this busulfan preconditioning regimen, and there's potentially a path to get to a more benign preconditioning regimen. Where do you stand with both of those efforts?
Yeah, I think the way I imagine the life cycle for Casgevy is that, at some point, a targeted conditioning agent is going to become a reality, and that's going to expand the addressable patient population significantly, 3 to 4x perhaps. But it's not easy to bring that, in a commercial setting. We still have a lot of work to do. But for both Vertex and us, this is a high priority. They have multiple agents that they're progressing forward with different targets. We have a c-Kit antibody that we acquired from a company called Magenta, and we put a different toxin on it to develop it specifically for sickle cell and thalassemia, where we want the agent to be relatively safe, so the toxin is not as toxic as you might in the oncology settings, and we want a very short half-life where it disappears before we put our cells in that are edited cells, right? And so what that gives you are multiple reasonable shots to get a target conditioning in play, but once we've established the market, established the infrastructure to get Casgevy to as many patients as we can. So it's going to significantly increase the value of Casgevy if we get a conditioning that can show chimerism even in NHPs. So let's see where the data land, but this is a high focus for us. The other part that we're working on, the 10-year solution is in vivo editing for whether the bone marrow or any of the hematopoietic cells. It's more complex than most people imagine right now. I think you're going to see upstarts saying, oh, we've figured out HSC editing. The thing is, you have to do HSC editing without editing everything else at doses that are tolerable, that also ensure durability, and they've edited the right cell types. But that said, I think we are trying a number of targeted LNPs where we decorate the LNPs with different factors that may give us this preferential selective editing. And so that is an effort that we're putting a lot of resources behind. It just takes time. But that's sort of the 5 and 10-year horizon if I think about what may happen with an autologous cell therapy associated with sickle cell and thalassemia.
On the in vivo platform that you commented on earlier, we're going to see, I believe, first data this year in cardiovascular disease. Could you just frame that for us, how to think about this first data set and what would be meaningful?
Yeah. We haven't guided to a time point for data yet, but we are accruing data. And, we have 2 trials ongoing for 2 cardiovascular risk factors that are well described in literature. One is LPA, and the second is ANGPTL3. And they both are independent risk factors. LPA, for instance, we just looked at the number of cases where people have high LPA. It's not patients, it's people. And some of them are in their 30s and 40s, and they get heart attacks, even though they're, very healthy in their lifestyle, they exercise. because they have significantly high LPA, and it's kind of genetic in a way. And what's been shown across all these population studies is lower LPA is better. You have lower risk of cardiac events. What hasn't been shown yet is whether pharmacological intervention, which reduces LPA, then changes outcomes for people who are born with high or low LPA. And we're going to get that answer in the next 18 months, hopefully, or next year, with the trials that Novartis and Amgen are running with ASOs and siRNAs, respectively. And so if you can show that pharmacological correlation of reducing LPA with better outcomes, we're starting off at a very different point in our development journey where those parameters have been established. And we can then say, why not do this with gene editing, which is a much better way of reducing this risk factor in a sustained fashion versus a sawtooth effect that you may get with an ASO or siRNA, and combined with the convenience of a one-time dosing. And so we're quite excited about those trials. I think, as we dose patients and collect data, we'll see what dose level we need to get to, what the therapeutic index is. But a lot of what we're doing is very similar to what Intellia did with HAE and TTR. So we'll see where our data land. But that's going to be a signal for us where if the data are positive, one, we're very excited about these indications, but we're also going to really turbocharge 6 other indications where we're going to use the same LNP platform.
You also have an emerging autoimmune vertical, and you're evaluating a drug in lupus. Perhaps talk to us, just given now how many assets are kind of entering this space in cell therapy, how you could be differentiated. And secondly, are you going to be able to take all these assets forward on your own, or are you thinking about partnerships on the forward?
Yeah, absolutely. I think while we see many cycles with investors and biotech ebbs and flows, there are these moments in time when you first describe certain data. I remember when the Emily Whitehead data, Carl June presented Emily Whitehead's case study, and it just changed how people thought about cell therapies in oncology. And this was Emily Whitehead was like 4 or 5 years at the time, and she was cured of a terrible leukemia. And so the same sort of thing is happening with the George Shett data for cell therapies in autoimmune. You've had patients with very severe autoimmune diseases that are completely in remission and durably with a single administration of cell therapies, in this case, CD19 cell therapy. So it just opens up an opportunity that's even larger than oncology, with an opportunity to address, bring something to these patients who haven't had a solution for a long time. I think we've all seen these TV shows where lupus is the underlying cause of everything that happens, and it actually is a very complex disease. You get all these cerebral symptoms, you get neuropathic pain, you get obviously kidney issues and everything else. And if you can have a CD19-directed cell therapy that's potent, that gives you deep B-cell depletion, I think it can be transformative. Now, the question is, there's like 20 agents in the space. There's autologous CAR Ts going into it. There's T-cell engagers, antibodies now, potentially ADCs, and then you have allogeneic cell therapies, both allo CAR Ts and allo NKs. So what's going to win? And I think this is why we are positioned to win this space. And this is sort of all these factors coming together that we couldn't imagine. We were looking at it saying, gosh, this is sitting there for us to win in this space. The reason is the burden of B cells you're going after is not as high as oncology. So with an allo CAR T, you can get full B cell depletion. Cell therapies go and deplete B cells in the tissue with the tissue-resident B cells that bispecifics and antibodies may not be able to. From a safety standpoint, the safety bar is high in autoimmune. You may not want to take any secondary malignancy risk that you may get with lentiviral-derived auto CAR T. So allo CAR T is better where it's CRISPR edited and much more directed insertion of the CAR. The safety profile from a CRS and ICANS standpoint also is better for allo CAR T. Allo CAR Ts are better than allo NK cells from a dosing standpoint and the ability to reduce COGS to a level that's meaningful commercially. And we as a company have the best allo CAR T with a commercial-ready manufacturing facility, the lowest COGS out there, and the ability to create hundreds of doses available for trials that others don't. So for all those reasons, I think we've been handed this opportunity where we want to take full advantage of it. We just announced the hiring of our Chief Medical Officer, Naimish Patel, who came from Sanofi, and he was leading their entire autoimmune franchise there and developed Dupixent from the early days with incredible experience in the autoimmune space. We built a team around him to help with autoimmune. And so this is very high priority for us. We're moving as fast as we can, and it helps that we have a strong balance sheet to support all the things we need to do to put us in pole position.
And so will you take all these programs forward?
Yeah, I mean, we have that the interesting, we have that flexibility. Given what we believe about Casgevy and the outlook, we have the flexibility to take both autoimmune and the in vivo programs forward ourselves all the way. Now, does that mean that we're not active on the BD front? I mean, I would say at JP Morgan this year, we had interest from at least half the top 20 pharma companies in these two programs, LPA and autoimmune CAR-T. And so we'll continue talking to pharma companies because we don't want to turn a blind eye to that. But we're not going to partner both. At best, we partner one or the other. But there is the option to take both forward ourselves, which would create tremendous enterprise value for us in the near term as we get to HPOC.
I want to get over to the oncology portfolio because that's one of the verticals you started with, and you've been optimizing along the way, and we're potentially going to see some data this year. Maybe walk us through what we could see from that effort and where you stand with the next generation assets.
Yeah, we're glad that we're making a very persistent effort in oncology because it does take that, the next generation to get there. When we started the first generation CAR-T in CD19, CTX110, it was incredible to us that you're getting durable responses. Some of the patients that had complete responses with CTX110 are cancer-free 3 years out. The gold standard for oncology to be disease-free is 5 years. And I'm sure we're going to get there with patients who had the original complete response with a single dose of an unoptimized allo CAR-T that we created using CRISPR. Now we have CTX112, which is much more sophisticated, and we made these edits to make them that much more potent, yet retain the safety features. And so with DLBCL, while the landscape has changed a lot, there's a few things that have emerged now, which is T-cell engagers are not as good from a durability standpoint as people originally imagined them to be. Okay? When people looked at 9 cycles of glofitamab, whatever, you said, gosh, at 15 months, looks like everybody's still in CR. So this could be the same thing that we're seeing with Yescarta, perhaps, where if people are in CR 1 year, they're probably in CR 2 years. And that hasn't been the case. They've lost nearly half the CRs between year 1 and year 2. And you're seeing that in the marketplace. You're not seeing bispecifics all of a sudden have a huge sales trajectory that people anticipated because of this durability reason. And the real-world data on CR is also lower than what people saw in the clinical trials. With autologous CAR-T, you still have a very high bar to beat. But what's also happened is only about 25% of the patients that are eligible or should benefit from this therapy are getting it because it's basically saturated all the academic medical centers, and all the community practices don't know how to deal with it yet, and they never will. This is too complex for them. There's reimbursement risk. There's inventory risk. There's risk of CRS and not being able to deal with that with the patient. So it's never going to get to that population. And the only way to get to that 75% of the population is to have allogeneic CAR-Ts with that durability. And so we'll have data this year in DLBCL. We've seen some other data sets recently from other allo CAR-Ts, but we'll have our own data set. You may not get the whole durability answer at this point, but you'll get a good indication of which way it's trending based on at least comparisons of 110 to 112 or what you're seeing with this patient population. So we'll have data later this year that informs not only our oncology path forward, but may also read through to autoimmune.
And just remind us the targets here. So we'll see CD19 and BCMA?
Well, CD19, with CD70 is the other program that we're advancing right now in solid tumors and in heme malignancies. We haven't said when we're going to have the data for that yet. But, with CTX130, which was a CD70 targeted allo CAR-T in heme malignancies, in T-cell lymphomas, we already saw data that was a product. We saw CRs in patients that had no hope. In fact, one of the patients was the subject of an NPR article where it was a former NBA player who couldn't walk because they were wheelchair bound because they had lymphoma all over their feet and all over their skin. And a month later, were dancing around, walking around, running. And you've had these cases in these very difficult to treat lymphomas, and CTX131 gives us that much better opportunity to treat these patients, get more patients in CR, in complete remissions. And some of the patients, we actually want to get into CR and go on to get an allo transplant, which is actually the best outcome they can get. So we'll have multiple data sets in addition to solid tumors that inform our our general cancer franchise strategy later this year.
And maybe just to round out here, the regenerative medicine vertical, how much are you prioritizing that effort with type 1 diabetes or anything else?
Yeah, it's a significant priority. Again, we have to make bets while others are maybe written it off or saying it's so difficult because that's how we're going to create alpha. That's how we're going to create tremendous value. And what you're seeing is we're already seeing a lot of benefit with the beta-2M edit with our cancer programs. We've made other edits like HLA-E knock-in. We've made edits like PDL1 knock-in to make cells stealth. And if we crack the nut on making these cells stealth, it opens up the possibilities not just for type 1 diabetes, but for many of the organ systems, right? I mean, you can imagine now stories where people are creating mini pig kidneys for human transplantation. They will all need these same edits around making them stealth. You're seeing islet cells is the best place to start because that's, you know, the best control experiment to see if these cells truly are stealth. And now we have both a device approach and a device-free approach that we're developing ultimately that's going to inform us on not just the stealth nature, but are we getting enough efficacy from an insulin production standpoint in a glucose-responsive fashion with these cells that allows the dose titrate. So the last part of this is now that we have the program back from Vertex, we're now doing all the development and manufacturing ourselves. Once we can do the manufacturing ourselves, it gives us tremendous flexibility to have multiple generations of products and move them all into clinic and parallel process different theses at the same time.
Well, with that, Sam, thank you so much. Really enjoyed the discussion.
Yeah, thank you very much, Eleni.